The 10th recent discovery on the list is that extensive post-transcriptional processing or editing of genes occurs by machines called spliceosomes. To explain this topic, some general background information is needed. This slide shows the generic steps of transcription of RNA from DNA and then translation of the RNA message into strands of amino acids that make functional proteins such as enzymes. The translation step is performed by a very complex machine called the ribosome that was discussed in discovery number 6. That machine uses molecules called tRNAs in the process of translation to make a protein. The tRNAs are at the heart of the translation process, and the process for constructing the tRNAs was discussed in discovery number 5. RNA also has other functions, including to serve as building blocks in the formation of some molecular machines, and also in regulating gene expression, as discussed in discovery number 9. Discovery number 10 is that in eukaryotes, something very complicated happens in the step of translating the RNA message into protein. Eukaryotes are organisms whose cells have a membrane-bound nucleus and other membrane-bound compartments. In eukaryotes, DNA sequences that form genes have many segments called exons, shown as the numbered boxes in this diagram, that code for portions of proteins. Genes also contain segments called introns, shown in green in this diagram, that do not code for protein segments. Recently, it was discovered that all the introns are cut out and that some coding segments are spliced back together to form the final RNA, which is translated to make a protein. Under different conditions and in different tissues, different combinations of exons are spliced together, so that many different proteins are constructed from the same section of DNA. Somehow the system knows which segments to splice together in different tissues and at different times. Because of this process, the number of proteins in a complex organism greatly exceeds the number of genes. The cutting and splicing process is performed by a machine called the spliceosome, and the process is believed to be governed by a splicing code. Commenting on this topic, Philip Ball, in his recent book, How Life Works, stated that there is at least as much regulation after transcription than before transcription. Remember that transcription is the process of making RNA from a DNA sequence. In discovery number 9, we touched on the newly discovered and enormously complicated world of RNA regulation of gene expression. Ball states that the regulation that happens after transcription, in the cutting and splicing of segments of RNA and also other editing processes, is as complicated as that involved in expressing or transcribing the genes. Two other authors stated that introns and alternative splicing are perhaps the biggest surprise in the history of molecular biology. Considering all that has been discovered in molecular biology, such as molecular machines, the digital information processing system at the core of life, and logic operations carried out by molecules, that is quite a strong statement. The statement demonstrates that this was definitely not expected by those who think from a Darwinian perspective. It's another example in a long list of discoveries in molecular biology that were shocking to those who believe that all aspects of molecular biology and of life came about by a mindless process. This slide contains some basic facts about this topic. On average, each human gene encodes about 6 different proteins. About 90% of all human genes are alternatively spliced, and this is especially prevalent in the human brain. 50% or more of genes are spliced differently in different tissues. So this is not a rare occurrence, but is typical for genes in humans. This is a very important phenomenon to understand because errors in mRNA processing are associated with cancers and neurodegenerative diseases. This image is taken from a review article on a protein called tropomyosin. Tropomyosins are considered a classic example of alternative splicing and have been extensively studied. These proteins act as molecular switches to regulate actin filaments involved in muscle contraction, as well as in the cytoskeleton. There are over 40 forms of this protein in humans. This image shows several variants that are produced in different types of cells. As shown in the full sequence of the gene at the top, the gene is composed of various segments indicated by different colors and different numbers. These colored boxes represent the protein coding sections or exons. The horizontal lines indicate intron sequences that are not used for protein production. A system involving a machine called the spliceosome processes the gene by cutting out the introns and splicing certain exons together to generate variants of this protein in different cell types. In discovery number 9, the discussion included the fact that in different cell types, different genes are turned on and off. The present discovery includes the fact that in different cell types, different proteins are made from the same RNA transcripts. Some proteins have even more variants or isoforms than tropomyosin. In mammals, a protein called neurexin, involved in neuronal development, has more than 2,000 variants. A gene called SLOW is used to generate proteins that form ion channels in inner ear hair cells that are important for distinguishing a range of sounds. In chickens, this gene has over 500 variants. A gene called TTN makes a protein called titin, the largest protein in the body, which is involved with proper functioning of heart and skeletal muscles, and is also present in the embryonic and adult nervous system. It has over 1 million variants. In humans, on average, there are 8 to 10 introns per gene. So in higher organisms, cutting and splicing of RNAs is not a rare occurrence, it's common. To my knowledge, the world record is held by the gene DESCAM. DESCAM aids formation of the complex architecture of the nervous system by providing a molecular code that enables neurons to distinguish between self and non-self. Aberrant DESCAM expression is linked to central nervous system defects and cognitive deficits observed in conditions like Down syndrome and autism spectrum disorders. The DS in DESCAM stands for Down syndrome. In a type of shrimp called the tiger prawn, there are over 21 million DESCAM variants. This image from a recent research article shows the entire DNA sequence. The black and colored vertical lines represent protein coding sections of the DNA with introns in between. All the black vertical lines are included for each protein, but only one red, one blue, and one green line are used in making a protein. In this way, an enormous number of combinations are generated. So genes should be understood as resources for making families of proteins. Since different protein variants are made in different cell types from the same RNA transcript, you're probably wondering how the choice is made as to which protein to make in different cell types, and how the alternative splicing process is regulated. It's incredibly complicated and certainly not yet fully understood, but some facts are known. I'll mention a couple of those facts and then encourage you to watch a few videos illustrating the process. First, the regulation of splicing involves associations of RNA with RNA-binding proteins. RNA-binding proteins were discovered relatively recently in 1986. In humans, roughly 3,000 genes code for RNA-binding proteins. Different sets of RNA-binding proteins are expressed in different cell types. The next slide will provide links to videos that describe the spliceosome, the machine that does the cutting and splicing. The image on this slide shows known consensus sequences that are involved in splicing. These sequences are recognized and used by the splicing machinery, as will be described in the videos. I strongly encourage you to take the time to watch these videos several times. You need to see this system in action to appreciate it. Without following all the details, you can still get a good sense of the complexity of this process, including the many different protein and RNA parts that must come together in specific ways at different stages in the operation. And also that the many parts must rearrange in specific ways at different stages. The dynamical nature of this machine is remarkable and unlike any machine that humans have ever made. To put into perspective the difficulty of trying to design this process, keep in mind that scientists are not yet able to even design an antibody sequence that will bind well to a viral spike protein antigen to neutralize a virus. This was demonstrated during the recent COVID epidemic. Considering that humans today, with all our directed intelligence in protein modeling, and with all our computational power, cannot yet design a protein sequence that will just bind to another specific protein, it's mind-boggling to imagine how the spliceosome system came about with all these different protein and RNA parts that managed to perform all these complex and precise manipulations. The main components of the spliceosome are referred to as SNRPs, which are composed of RNA binding proteins and an RNA strand. The SNRPs bind to specific sites on mRNAs due to base pairing by the RNA strand. The author of this article in 2003 suggested that the spliceosome may be the most complex macromolecular machine in the cell. And that's saying a lot considering the complexity of the ribosome that was discussed in Discovery number 6. This article points out that nearly 300 distinct proteins and 5 RNAs are involved in splicing. The article also points out the dynamical nature of this machine, which was illustrated in the videos. A section in the article explains why so many proteins are involved. One very important aspect is that multiple proofreading steps are needed. splicing must be very accurate. The article states that each splicing event is inspected multiple times by multiple different factors. Proteins are also involved in tagging the final RNAs as having been spliced, which regulates the fate of the mRNA for such things as export from the nucleus, translation into protein, and for localization to specific places in the cell. Finally, adapter proteins link the spliceosome to the RNA polymerase so that the two processes are intimately connected. In his book, How Life Works, Philip Ball states that the spliceosome is like no machine that we humans have made. Some of the proteins seem to be constantly changing, coming and going, or rearranging as the assembly does its job. Today, humans can design supercomputers and spaceships and nuclear submarines and such. But this machine is light years beyond what we could design with our molecular biology techniques and knowledge at the present time. That should give you an idea of how sophisticated and remarkable this machine is. The notion that this was built by a mindless process of random mutation and natural selection is, in my view, inconceivable. The spliceosome is clearly an irreducibly complex machine. But while it is absolutely amazing, it's also important to note that it's part of a complex, higher-level system that must be coherent in order to accomplish the task of correctly cutting and splicing RNA transcripts to make different proteins in different cell types and for different developmental stages. I've mentioned several aspects of this higher order system already, but of course the splicing code itself is at the heart of that system. As was shown in the videos, introns and exons contain additional sequence elements beyond the consensus sequences that either repress or enhance the process by binding to proteins or RNAs. Deep learning AI and other approaches are currently being used to try to sort all this out. Understanding the splicing code is tremendously important because, as I stated earlier, errors in mRNA processing are associated with cancers and neurodegenerative diseases. This very recent review article states that a major goal is a splicing model that can predict from genome sequence alone the complete set of transcripts and their frequencies in any cellular context. This slide shows a few additional references beyond those already cited. To summarize, discovery number 10 is that extensive post-transcriptional editing of genes occurs. Here, only one aspect of that was discussed, the cutting and splicing of transcripts by machines called spliceosomes. That process involves a splicing code, the nature of which is still very far from understood. Since experimental evolution studies haven't shown the appearance of even very simple new molecular machines or integrated functional systems, as discussed in discovery number 7, and since the mechanism of random mutation and natural selection is known to have severe limitations, as also discussed in discovery number 7, RNA editing by the spliceosome and the splicing code seem to be enormous informational discontinuities, a level of information that is far beyond what unintelligent processes can accomplish.